Okay, let's get started. So welcome everyone to the Population Genetics workshop for this year. We're going to start off with basic principles and concepts. Okay, so those of us in genetics would understand the concepts and haplotypes. But for those of you that don't, let me just get my pointer. For those of you that don't, we have the slide over here depicting SNPs. So that is in the vertical box between individuals sequences, you'll see that there is a variant that's polymorphic. So that is a single nucleotide polymorphism in a SNP. And those that are inherited together, are called haplotypes. So SNPs that are consistently inherited together or found together are thinking about software, are found together are called haplotypes. These haplotypes can be of varying sizes. It could be of SNPs right next to one another. It could be SNPs that are not right next to one another, that's in close proximity to one another as well. Okay, so we all should understand the concept of genotyping and sequencing. So whole genome sequencing is about 300 million bases. It says here 100% of the human genome, but we know that that is not true. There are portions of the human genome that are not sequenced. And a snapshot of this, so all the protein coding portions of the genome, if you would sequence those, so that's whole exome sequencing. So that's about 60,000 bases, depending on how diverse the exome is, and that's only about 2% of the human genome. So you're only getting a very, very small fraction of the genome in that information. Whole exome sequencing is routinely used for more clinical applications. So with there, you're really interested in finding variants that are going to have a direct impact on protein production or protein formation. So for population genetic practices, we don't use whole exome sequencing just because there's too little information available to us, and generally the distance between SNPs is too great, so we cannot do a lot of the analyses that we do. So we don't use whole exome sequencing. What we routinely use, and that's partly because of the expansiveness of the data, but also for cost reasons, which I'll get to just now, we generally use large-scale genotyping. So initially, you were looking at about 1 million bases with the small-scale arrays back in the day. Let's put it that way. But now you're looking at arrays that can give you close to 3 million SNPs. So you're looking at dense information, and that spans from your autosomes to your sex chromosomes, even to your mitochondrial DNA as well. So you really are getting expansiveness of data. And depending on the number of bases, obviously, will dictate the percentage of the human genome that it covers. In terms of costing, the whole genome sequencing is very much dependent on the coverage that you have asked the sequencer to give you. So coverage meaning how many times is your SNP covered by the sequencer. The more times it's covered, the more sure you are that that SNP is there. So if you only cover it 4 times, the chances of it not being there, but it being sequenced, quote unquote, is quite large. But if you sequence it 30 times, you're more likely to know for sure that that variant is or isn't there. And as you can imagine, if you cover a SNP, the more expensive it gets. So if you are looking for low coverage sequencing, which is about 4x, you are looking at about 2,500 Rand per sample, which is quite economical, considering that the large-scale genotyping per sample will cost roughly similar. So you're looking at how much information do you want, but how reliable of the really worried about how many times a variant is covered, as long as it's covered, I would say at least 4 times. When you're moving on towards the clinical realm, you're looking at about 10 times that you want, just to be sure that any clinical genotype-phenotype correlation that you do find is in fact there. Okay. Okay. So let's talk a bit about the reference genome. So a reference genome assembly is a nucleic acid sequence that's assembled as a representative example of the genome in one individual organism or species. And it's used as a guide on which genomes are built. So you have sequenced 10 individuals, and you need to know how to put together the bits of information that the sequencer has given you. And to do this, you use a reference genome. Through a process of alignment, you are able to fit these puzzle pieces together based on this reference genome. So the accuracy of this reference genome is really important. It also helps with de novo assemblies. And you'll see later on in population genetics, what it really helps us do is it makes sure that we are always looking at the forward or reverse strand. And this is important when you are merging data. You don't want to be merging forward strand with reverse strand. It's either going to mean that you get complete junk out, or it means that you're going to lose the variance, a large proportion of variance, because it's not the forward and reverse strand are not aligning together. Okay, so there are different versions of the reference genome, and it gets continually updated. We are on, at the moment, GRCH 38, and a lot of programs use GRCH 38, but the previous version is also quite well utilized. It was one of the versions of reference genome that had least amount of problems, hence why everyone is hanging on to GRCH 37. There are also inherent problems with the reference genome, no matter what version, and that is that it's Eurocentric. You can imagine that the diversity in the world, we want to make sure that the reference genome that we have is encompassing of that. The reference genome that we have currently is from one man in Buffalo in New York City. I mean, how representative is that? So we want to make sure, and there are endeavors out there to do this, that the reference genomes that we have are representative of the diversity. We have quite a few projects in-house, also in H3 Africa, that is attempting to address this by constructing reference genomes for African populations. And this is particularly important in the clinical genetics realm, where you have a variant that you suspect is disease causing, but you need to make sure that it's not a common variant. And what databases do you have access to? You have access to European databases, and that's not going to be particularly informative. And the same can be said when you're aligning your sequencing to a reference genome. You can imagine that there's a lot of errors that could occur in this alignment process if your reference genome and your sequencing reads are not from the same population either. There are also gaps in the reference genome. You can see in the image on the right-hand side that the gaps are indicated by triangles, centromeres in red, translocations that looks like navy, and inversions in orange. So you can see that there are still quite a few gaps in the genome. About 5% of the genome still has gaps. So there's a lot of work that's currently happening to fill in those gaps. Okay, so let's move on to some measures of diversity. So when we talk about minor allele frequency, so that is, or MAF, that is the frequency at which the second most common allele is present in a sample set or population. So that could be within your particular file that you have of individuals. That could be within a population in a whole. So if you look at an online database like Genome AD or 1000 Genomes, you will be looking at minor allele frequencies based on either that whole data set or on population specific. And obviously, if you are interested in major allele frequency, that is also available as well, but not as informative for diversity purposes. So a statistical manipulation of the MAF is the fixation index. And this measures the level of population differentiation due to population structure, which has been caused by differences in allele frequencies. And the higher FST value, the larger the degree of separation between populations. So you can imagine if you are looking at a European population, you're looking at an African population, you'll have a relatively high FST value. When you're looking at African populations specifically, some of the FST values, when you compare, for instance, East African population to a West African population, is as much as if you were to compare European population to a West African population. So the diversity within Africa is vast and comparable to the diversity between Africa and other continents. So there are other measures of diversity. Population branch statistic, levels of heterozygosity, you can go on and on and on. Every researcher has their preference of measures of diversity, and each statistic or estimation has positive and negatives. Okay, let's speak a bit about linkage disequilibrium. We are hopping around a bit in terms of topics, but we're trying to cover everything that we are going to be talking about in the lectures. So if there's no flow to the slides, I apologize, but we're just aiming to cover everything. So linkage disequilibrium is the non-random association between alleles of genetic loci. They're now in close proximity to one another. So positive LD exists when two alleles occur together on the same haplotype. Remember, we went through the concept of haplotype. And there's positive LD if this is seen more often than expected. Negative LD exists when two alleles occur on the same haplotype less often than expected. So you can imagine the closer SNPs are, haplotypes are, the more likely that there is LD between them. And as the figure on the right shows, it depicts linkage disequilibrium within a population. And in the initial generation, which is the first block on the top, you'll see that there are quite large LD blocks, which is depicted in the various colors. And after successive generations, these LD blocks decay. And this is because of admixture and recombination events. So after 1,000 generations, you'll see that there are extremely short LD blocks. So this, you can imagine, as a measure, could be used as a measure to calculate the date of admixture, right, depending on LD block size. Okay, so we mentioned admixture. So what is admixture? Admixture occurs when two previously isolated populations interbreed to form a new population, multiple ancestral sources. So you can see population A on the left and population B on the right. First generation admixture, again, you can see those blocks are relatively straightforward. Recombination has occurred. After a few generations of admixture, those ancestral blocks will be a lot smaller. Okay, so we're moving to a bit more practical aspects. So during the workshop, we will be working with a few files. So the two main file formats that we will be using are binary plink files. So you'll see that the file extensions at the end will end in .bed, .bim, and .fam. So these three files are generally used for housing genotyping data. So you can house just a few individuals in these types of files, all the way up to thousands of individuals. And obviously, the more individuals you have, the larger the files are going to be. And it can be pretty tricky when you have thousands of individuals if files are extremely large, although this file format does a good job of condensing this information as much as possible. Okay, so a .bed file, you'll notice that if you try and open it up in your terminal, it'll ask you, it's a binary file, do you want to see it anyway? And you say yes, and it's complete garbage that you see. So don't worry, and there's nothing wrong with your file. It's just in a format that is not human readable. And that is one of the ways that what they've done to decrease the file sizes. The .bim file has your marker information, so you can see the screenshot on top left. That is a screenshot of a .bim file, actually the one that you're going to be working with. So you can see in the first column you have your chromosome number, your RSID. The third column generally will have your centimorgan, so your rate of recombination, your recombination rate in that site, the base pair position, and the two alleles at that site. Sometimes your files won't have RSIDs, so generally it's good practice to annotate your genotyping files with RSIDs, and RSIDs is just a number given to that particular site, that particular variant, to ID it. And you can then go into dbSNP and put in the RSID, or you can just Google the RSID, and it will give you a lot of information about this particular variant, including allele frequencies in other populations. It'll tell you whether this variant has been associated with any phenotypes or any diseases. So that is one way to make sure that that particular SNP has a unique ID compared to all the others. You may find in some of the BIM files that you will, rather than RSID, you'll have the chromosome number. So in this case, 1, you'll have a colon, and then you'll have the base pair position. So that is particularly for variants that are that don't have RSID, and in that case, it'll be novel SNPs. So SNPs that are not found as common as the rest of the SNPs, and they just have not been assigned an RSID. And those SNPs in population genetics are actually quite interesting, because it means that generally they are unique to the population, or can be unique to the population. Okay, so a FAM file is the screenshot on the right. You can see here, if you have done a bit of work in population genetics, that you'll notice HGDP. So during the workshop, you'll be working with HGDP data. That is Human Genome Diversity Project data. It's a consortium that aims to genotype large-scale worldwide populations, and they actually have recently brought out high-coverage whole genome sequencing data as well, which is very useful. So again, we're going to be working with that data, the genotyping data, not the sequencing data, during the workshop. Devin will be putting those files on the chat if they are not on there already, and those will be the files that we are taking through the workshop straight from the beginning to the end. So a bit about the FAM file. So the first column, you'll have a family ID. So you may have individuals that are on the same family, and you will then tell software that you are using to manipulate these files that these individuals are on the same family. Then you have your individual ID, unique individual ID, and the rest of the 4 columns can be information regarding the biological sex of the individual. You can annotate with case control association, case control assignments, and you can also put in other family information as well, if there are cousins or grandparents or whatnot. Okay, so the second file that we have is the variant call format. You'll see the screenshot on the right-hand side of a variant call format file, and this file is generally used for sequencing projects. I've very loosely say it's the same information as in the Plink files, but it's packaged differently. Okay, so you will still have all the information regarding your SNP, your mark information. You'll have information regarding the individuals. The only information that you won't have in a VCF file is information regarding relatedness, so where the individuals are related to one another. That is one of the only differences. But that doesn't mean to say that you can't put it through software to determine relatedness. You just can't house that information in the file. With the VCF, you will also have information regarding the depths of coverage. So again, going back to coverage, how many times was that specific SNP covered in a sequencing reaction? It'll give you a score, reliability score, in terms of how likely is this variant to be a true variant, and you can then filter your VCF based on that to get high quality data as well. Okay, so that is all the principles and concepts side of things. I'm just going to open up the chat to see if there are any questions. If you have any questions, I'm going to wait 2 minutes for you to type them in the chat, and then we are going to move on to Dana's portion of the lectures, and she'll be giving us a lecture on genotype data quality control and merging. I've given her an extra 15 minutes, so she can talk a little bit more. Okay, so we have some information about installing Plink. And Devin has sent an email with a link to all the files in OneDrive. Again, if you can't access it, please let me know, or please let us know, so that we can address that. Okay, so I'm going to hand over to Dana. I'm going to stop sharing, and Dana will start sharing on her side. Thanks, everybody.
Okay, online people, can you hear me? Just give me a little thumbs up. Sorry, you're going to see my whole screen. Okay, cool. People can hear me. That's nice. Okay, cool. On a second note, for all the people who have Windows and have not figured out how to install Plink, hit me up and then I'll help you during the tea break, because I figured out how to do it, but we'll do that after the tea break. Sorry, I was like, why can't I share my screen? And I'm already sharing. Okay, cool. So today we're going to talk a little bit about genotype data quality control and merging. Okay, and Caitlin already took you through what genotype data is. So hopefully you understand its formats, where it came from, before this lecture. Okay, so we're going to talk about genotype quality control. So why do we need to do it? The steps of genotype quality control, and then we're going to take a brief look at Plink and why we are using this weird software that we've had such trouble installing, and why we put ourselves through this hell to install it in the first place. Okay, and then I'm also going to briefly talk about merging. Again, why do we need to do it? The steps of how to do it, and then common issues. And then we're going to do a little demonstration after the tea break on all of these different topics. Cool. So the first question I'm going to pose to the audience: why do you think we need to perform quality control, or QC? Like, why do you think we have to do this? Anybody? If you're online, you can just type it in the chat. If you're in the room, you can put up your hand. Yes, Crystal. Exactly. Perfect. Okay. Basically, raw genotype data is inherently imperfect. And the reason why is because sometimes your DNA samples don't have great quality. Sometimes you have poor DNA hybridization to the array. So Caitlin took you through briefly like what an array is and how we get genotyping data from an array. Sometimes your DNA doesn't actually stick to the array properly, resulting in genotyping errors. And sometimes, as wonderful as we are as humans, we can also mess up. And sometimes we can mess up samples or contaminate samples. And this is our fault. And we need to control for this. Otherwise, we're going to get problems. So quality control aims to identify these errors and remove these technical errors before we move on to our next steps. And this is because technical errors can seriously mess up our downstream results. And I've given an example here. So it's from a 2010 paper. This paper was initially published in Science, very fancy journal, where they found this awesome result. And then afterwards, they actually went back to their original data and they discovered they didn't perform thorough quality control. And when they did and redid their analyses, they actually didn't find that result. So that was what we call a spurious association, or like a false positive result. That result was actually not true, and it was just a technical error produced during genotyping. Okay, and then they had to retract the article and look a little bit silly in front of the whole scientific community. Okay, so we don't want to do that. We don't want to look silly and publish weird results that are not true. So we want to perform quality control. So some common steps of quality control is that we want to exclude SNPs that are missing in a large proportion of samples, because that can also lead to spurious associations. And we want to exclude samples that have high rates of genotype missingness. So some individuals may not have been genotyped for a particular SNP because that DNA did not hybridize to the array properly. Basically, in genotyping quality control, we want to remove any missing data because that's going to be funky with our analyses. Okay, we want to check for discrepancies between the reported sex of individuals and the sex based on their genotyping data. It's going to be very awkward if you get an individual who reports as male, but then their genotype data indicates that they are XX. Okay, that's a bit weird. Obviously, in our day and age, anything can be true, but normally this is an indication of sample mix-ups. Okay. And then also you want to exclude monomorphic SNPs. Okay, so these are basically where the minor allele, do you guys know the difference between the major allele and the minor allele? Okay, I see lots of nodding, so that's good. So the people who, when the minor allele is not present in the sample set, that means everyone has the major allele, right? Everyone has the reference. So it's generally not informative for our analyses because normally the allele we're looking for is the alternate allele. Okay, and this is an important little calculation I need to take down before we get into our demonstration, is we need to calculate what is the minor allele frequency of these monomorphic SNPs. Okay, someone online said, may you please explain the difference between the major allele and the minor allele? So the major allele is the reference allele, so what the reference genome says you have. And then the minor allele is the alternate allele, or if you have a polymorphism, or if you have a mutation at that site, you won't have the reference allele, you'll have the minor allele or the alternate allele. Okay, so does that make sense? Just indicate with a little thumbs up. I just did that, sorry. Yeah, cool. So major allele is the reference allele, so what your reference genome says you have. The minor allele is any allele that's not the reference allele, so any little mutation that you've got. Cool. And then this is so we need to calculate what is the minor allele frequency of any monomorphic SNPs. It's this calculation over here. So 1 divided by 2n, where n is the number of samples. So if you have 60 samples, it'll be 1 over 2 times 60, which is 120. And then you work out what the calculation is. Okay, thank you for the explanation. No stress. Cool. Okay, so these are the first little bit of quality control steps. Some other steps are you want to exclude SNPs below a certain minor allele frequency. So this is kind of different to filtering out your monomorphic SNPs. This is, for example, if you don't want to look at any very rare variants, if you only want to look at common variants in your analyses, you want to just filter out those rare variants. So anything below a certain threshold, and then the researcher can specify whatever threshold they want, essentially. So if you only want to look at SNPs that have a minor allele frequency greater than 10%, you can set that, and you can remove any other SNPs that have a minor allele frequency less than that. You want to exclude SNPs that have deviated from Hardy-Weinberg equilibrium. So do you guys know what Hardy-Weinberg equilibrium is? I think that's a whole other can of worms that we're not going to go through today. But basically, it's an indication of population evolution and sample evolution. But in our cases here today with genotyping quality, it's normally if your SNPs deviate from Hardy-Weinberg equilibrium, it's more likely that this is a genotyping error as opposed to your populations evolving at a really, really high rate. Okay, so we just want to filter those SNPs out because they're probably just a genotyping error. And then also you want to double check which version of the genome your data was aligned to. So Caitlin explained in a previous lecture, there are a couple of different versions of the reference genome. So we work with GRCH37, but there's also GRCH38. There's other reference genomes, and we have, like, I'm sure you guys may have heard of the telomere to telomere project, which is the newest reference genome of, like, without gaps and all that stuff. So you want to double check, you may have different data sets, right? Or you may be wanting to perform imputation, or you may want to merge these different data sets. And you can't merge data sets that were aligned to different versions of the genome. I like to say it's kind of like we're comparing fruits. So they're all reference genomes, right? They're all human reference genomes. They're all fruits. But we want to make sure we're comparing apples and not apples and oranges. Okay, you can't compare GRCH38 to GRCH37 because the coordinate sites, like the actual positions, are going to have slightly different numbers. Okay, slightly different variations. So that's really, really important before we want to perform imputation and before we want to merge. Hey, any questions so far in the room or online? I'll give a little moment before, okay, everyone seems good. And then why are we using Plink? Because it was a real mission to download. But we're using Plink just because it's a tool used for genotype quality control, among other functions. So it can also look at zygosity, it can perform GWASs, so it's quite a useful tool. We want to use it because it's open source. So you guys probably noticed you didn't have to pay anything to download it, which is awesome. It can be used by Mac or PC or Linux in theory. We're trying to figure out the downloading and the installation. So I know it doesn't feel like that right now, but it can. It's also very computationally efficient, and it can manipulate large data sets really, really quickly. And it's very easy to use. Okay, which it doesn't feel like it right now. I'm sure if you've never used it before, but when we get to the demo, I can show you it will be. So that's basically why we're using this tool. Okay, so any questions on genotype quality control before I move on? Yes. Exactly. For the people who didn't hear online, Plink does something. So Caitlin explained to you a VCF file and Plink binary files in the previous lecture. So the binary files are kind of, for lack of a better word, I'm just going to say like a compressed version of the VCF files, which is why Plink is able to run so quickly, because it's kind of compressing the data and able to move through it so fast, as opposed to going through a VCF line by line by line by line if you have a million lines. Okay, cool. XK. That's a good question. So the threshold for Hardy-Weinberg equilibrium, again, this is something that is set by the researchers themselves. So you can decide, so Hardy-Weinberg equilibrium works off a p-value. So it's kind of like the probability that the SNP deviates from Hardy-Weinberg equilibrium, that it's showing that a population is evolving. So in our population, or in our cohorts, or in our, what we're going to work through in our demonstration now, we use a threshold of p is equal to 10 to the minus 5. So that's 0.00001. Okay, but again, the researcher can choose in this case, obviously given in mind that work through. Sorry, I'm just reading the questions. Isn't minor alveolar frequency 0.1 too high if we are doing demographics? Yes. So this is exactly it. Yeah. So in our cohort or in our demonstration today, you're going to notice I don't filter out any SNPs below a certain minor allele frequency except for the monomorphic SNPs. Okay. So if you want to look at rare variants, you can. So the minor allele frequency is kind of like an optional extra if you want to filter out data or not. And again, minor allele frequency, you can choose the threshold. I just included that threshold there as an example. Cool. Any other questions? I think we are all good. Nice. Okay, someone also answered in the chat. Okay, cool. So in that case, I am going to run through merging now. So let's imagine that we are scientists. We've got a couple of different data sets. We've just quality controlled these data sets so they're nice and clean. And now sometimes we're going to want to merge data sets. So let's say I've got two different data sets, one from one population and one from another population. Why do you think we're going to want to merge them together? Any guesses? Crystal. Perfect. Yeah, that's a good example. So you don't have to repeat things for separate different data sets. Someone said comparative data analysis. Yeah, population 1 versus population 2. That's excellent. So it's a bit like what we're going to do later or tomorrow, admixture analysis. If you want to look at your population compared to a reference population, you're going to have to merge them. And then also a very simple answer, if you just have like separate data sets and you want to combine them to make a bigger data set. Okay, that's something called a mega analysis. We will want to pool raw data across different studies. Let's say you are a large consortium, like a Kappa consortium, or let's say you are a very, very big project and you've got lots and lots of money and you want to genotype millions of different people across the world. You're not going to do this all at one center. You're going to do it in batches, probably at different centers. But at the end, you're going to have to combine all of these batches together. That's another reason. And then, as I explained earlier, tomorrow, Yolandi is going to take you through admixture analysis. And in this kind of analysis, you have to combine some reference data, so like a reference population, to your target population. Okay. So that is an example of why we would want to merge different data sets. I see a question on Heidi Weinberg equilibrium on Teams. I'm going to get back to you after merging. Okay. So some basic steps on merging. We should perform quality control on each of the separate data sets. Okay, so we want to make sure that each of our little data sets are nice and clean before we put them all together. Otherwise, we're just going to be combining mess with mess and get a bigger mess. Okay, which is not what we want. We want nice little clean data sets to combine them together to get a pretty clean big data set. The second thing, which is really important to note, is that you can't just merge all of the SNPs across all your different data sets together. Okay, you can only merge those SNPs that are common or intersecting across all the different data sets. So I've indicated here on a little Venn diagram. Let's say you've got 3 different data sets. They all obviously have lots of different SNPs, but the ones that we can merge together are the common SNPs, which I've highlighted in pink there. Okay, those are the intersecting ones. Those are the SNPs that are present in all 3 data sets. Okay, and this is because remember in the beginning of the genotype quality control lecture, I said you don't want missing information. If you combine SNPs that are on one data set but not on the other, you're just going to add missing information, if that makes sense. Cool. So in order to do this, you want to identify what those intersecting SNPs are. And you're going to want to take them out of the data sets, basically, make sure that you keep them. And you're going to want to merge those ones together. And then just for sanity, we're going to want to do another little round of quality control on the merged data set. So again, we can filter for genotype missingness, individual missingness. We can filter again for Heidi Weinberg. This is just like a sanity check to really remove any other last residual remaining genotyping errors that may have been left in. And then I put a little asterisk next to this last step. You may want to identify and remove individuals that are related or individuals that are duplicated. For example, if you're combining two different data sets that were performed by two different studies, one individual may have been genotyped in both studies. You probably want to remove that individual because you're just duplicating the data. Sometimes you want to remove related individuals because these can also relate to, introduce spurious results downstream. But I put a little asterisk next to it because it depends on what next step is. Okay, if you're doing something like imputation, which we are going to do today, you want to leave in your related individuals because that actually improves the accuracy of your imputation. But if you're performing something like admixture, you're going to want to take them out. So we're just going to leave them in for now. And then removing related individuals will be Yolandi's problem tomorrow. Okay, so those are the 6 different steps for merging data. Very, very easy, right? Just 6 steps. It is easy. Cool. So when you're merging data, you may come across some commonly found issues. And these are the 2 most common issues I could think of. So maybe you have a low genotype rate after merging. This normally is because you didn't merge only the intersecting SNPs. Okay, you merged all of the SNPs together. And then when you perform quality control later, you're going to be given a big shock because when you screen for SNPs with missing information, you're going to be having a lot of SNPs removed. Okay, and that frightens everyone, but that's normally an indication that you didn't merge only the intersecting SNPs. When you merge the intersecting SNPs, your genotype call rate should be quite high, or as high as you need it to be for your threshold. So that's your first issue. And then the other issues are different merge failures. So the first one is maybe one data set was genotyped or aligned to one reference genome, like HG19, but the other data set was aligned to a different reference genome. Okay, remember you're merging apples and oranges in this case, which you're going to get a fruit salad. That's a big mess. Okay, so you want to merge apples and apples, so you're going to have to convert one of them to the same genome build so that you're comparing apples and apples. And then strand flip errors, which I'm not going to go through today because Plink has a really nice little documentation that goes through exactly how to resolve these errors. But it's a little bit similar to when your data sets aren't aligned to the same genome. Okay. And then one special note, a special consideration is merging genotype data and whole genome sequencing data. It seems like this would be easy, but it's also got some technical difficulties, which I'm not going to go through today because we're not working with whole genome sequencing data. But I have inserted this article here for you guys to read in your own time if you do plan on merging genotype data and whole genome sequencing data at some point. Okay, and I have lots of recommended reading for you, just on lots of papers that give you like a step-by-step process of how to conduct quality control. Some papers are why you want to do certain quality control procedures, and I'll make these available to you guys afterwards. And then the same thing for merging. And then, yeah, any questions? I'll make these slides available to you guys after the lecture, so you can just click on the hyperlinks for these papers. Any questions? Okay, there was one question on Hardy-Weinberg that I'll go back to. Cool, let me just exit so I can see that question. Where did it go? There we go. What could potentially happen if one has a high Hardy-Weinberg equilibrium threshold and it is recommended to have it higher? So I think if I understand correctly, it means that you've applied one Hardy-Weinberg equilibrium threshold and it's removed a lot of SNPs and you find you need to be more strict. I don't know if I understand the question correctly. One has a high Hardy-Weinberg threshold and it's recommended to have it even higher. I guess if it's recommended to have it even higher, you can definitely a lot of genotype quality control, I think when we do it, is a lot of trial and error. We see how many SNPs get removed, and we see if we're happy with the amount that gets removed. If we find we're being too stringent or not stringent enough, you can always adapt the number of SNPs that you remove, and you can adapt these thresholds. Is that correct? Yeah. So if you find you have a very, very strict Hardy-Weinberg equilibrium threshold and it removes a lot of SNPs, it could be an indication that something's wrong, but it could also be an indication that you're being a bit too stringent, and so you can relax your threshold to remove fewer SNPs. Does that make sense? Yes, exactly. Good point. Depending on the application that you are going to do next. So depending on what your next step is, yeah, you may want to be more strict or less strict. If one performs quality control per population, then merges the population, do they have to perform quality control again on the merged data? So I do this as a sanity check. Often when, in my experience, when I have done it, not a lot of SNPs are removed. Okay, there's not a lot of data left to quality control, but I do it as a sanity check. One, to make sure that my merge has worked. If you see you apply your quality control filters and a lot of SNPs are removed, that is often an indication that something's gone funky and you need to go back a step and redo it. So I do it as a sanity check, but often you'll see in our demonstration now, quality control metrics after a merge, because your data sets are nice and clean when you put them together, it doesn't move a lot. And if it does, then that's probably a problem and you need to go back a step and try things again. So it's good practice to do, basically. Cool. Alrighty. I also finished quite early, so if there are any more questions, perfect. Cool. Nice and slowly. Perfect. Okay. For the demonstration, you're not going to have Plink installed. You're just going to watch me. Okay. And then during the tea break, I did find a cool little way online to work Plink on Ubuntu. So if you guys are still on Ubuntu and having problems with Plink, we can sort that out then. Yes. Yolandi, do you want to answer this question?
In its individual. So it might not, a mixture kind of works, the algorithm itself works that way, but contact related individuals. So it's kind of like first when you see how many ancestral populations contribute to your admix or your cohort, you first want to make sure that it doesn't see it as one individual and just separate them as well. So when you map it to or like merge it and compare it to the reference, you want to make sure they're separate. Okay.
Excellent. Cool. Someone online also said, please help with installing Plink on Windows. We will during the tea break. You don't need to worry about Plink for the demonstration. You can just watch me. Alrighty, cool. Then I think if there are no more questions, I'm going to share my screen again so that we can move on to the demonstration. Okay, like I said, any installation queries or anything like that we'll deal with after the demonstration. So first things first, I hope you guys all have managed to at least make a little folder for yourself for this workshop, because we are going to give you data and you're going to be generating your own files, and we're going to give you like some codes, or maybe you're going to want to take down some notes. So I definitely recommend making a little folder in a place that's easily accessible for you, where you can put all of this. You can see I have one for all my software, and then I have another one for all the data that we're going to be working with. Okay, ignore all the files that are generated now. But so we have two different data sets that we're going to work with today. One we have called the reference data set, and one we've called the target data set. So as Caitlin mentioned, I think in the last lecture, we're working with data from the Human Genome Diversity Project. And just want to give you guys a little look at what we're working with today. So we're working with, you can see these are what we call the sample IDs. So each individual that's been genotyped, they get given a unique ID, which you can identify their sample by, and all the populations that we're going to work with. So you can see we have a lot of populations. Bigger. Better? Better. Good. Okay, cool. So these are all the, so yeah, again, sample ID. And then we have the different populations that we're going to be working with. So our reference populations today are several different African populations that are from the Human Genome Diversity Project. So Bika Pygmy, Mbutu Pygmy. We also have a French population to add some European ancestry into the mix. We have Yoruba from Nigeria. But most importantly, our target individuals are these little guys here. Okay, there are 14 target individuals. You'll see there's San and Bantu from South Africa. Okay, so are you guys familiar with the San population? Good. I see some nods. Okay, so the indigenous population in Southern Africa, and we have a Bantu-speaking African group. Okay, these are the populations that we work with a lot in our host gen group, and these are the populations that are really, really interesting to analyze. Okay, everything else are reference individuals. So you can see, I hope Devin will make these available to you, or you have already, these files. Okay, so you guys in an email, is that correct? Okay, there should be a link onto a OneDrive folder where these 6 files are. Okay, sorry, 7 files, because including the population information. Okay, you've got your reference file, and you've got your target files. Plink needs all 3 files for each, okay? Plink needs the FAM file for the family information or the individual information, the BIM file with the SNP information, and the BED file, which is the binary file. Okay, you need all 3 for each of them, otherwise things are not going to work well. Okay, cool. So you don't have to worry about getting that now. You can get that during the tea break. I'm just showing you guys what we're working with today. So the first thing I want to do is I want to move to, for those of you who are new to command line, I want to move to the directory where my data is. Yes. Zoom in. Okay. I can't zoom in. I can make it bigger. Does that help? I can't zoom in. Oh, I can zoom in here. Better? Okay. Good. People online, I'm sorry, this is as big as it gets. Okay, cool. So you can see here, this is the directory where I have my files. If I want to execute Plink, put a little dot and a / in front of any software you want to execute. And then you could see my software was in the other file. So I'm going to just take a step back, move to my software directory, go to Plink. And then if I want to see that Plink is working, I can just type version or anything just to make sure that things are working nicely. Okay, so you can see I've got Plink version 2 installed. Okay, so the first thing I want to show you is I'm just going to show you quality control on the reference file. Okay, I'm not going to show you on the target file. You guys can do that on your own. Okay, so the first thing we want to do, what's the first step in genotype quality control? Who remembers from our lecture? Exactly. Exclude proportions with exclude SNPs with a large amount of missingness. Okay, so we're going to do that. So with Plink, we need to specify we're using the software Plink. We need to specify also which files we are using, and that's why we say --b file. So we need to tell Plink that we're using the binary files. Okay, which files we're using? We're using the reference files. Make sure you take out the little dots there. Okay. Happy with this so far? So we're telling the computer I want to use Plink software. I want to use the binary file format. And I want to use this reference file. Cool. Happy days. Cool. The next thing that we want to do, as Crystal said, we want to have a large amount of genotype missingness. Okay. And so for our purposes, I'm going to say that I want to include SNPs that have a genotype call rate of 95%. Okay, so below 95%, I want to get rid of, essentially. Okay, so that's just a little technical thing. But basically, the flag for filtering out genotype missingness in Plink is --geno. Okay, and then we want to state what threshold we are using. We kind of have to do it backwards in Plink. So instead of saying 95%, I must do 100 minus 95, which is 5%, but we must put it in decimals. Okay, so is that 0.05? That's right? Sorry, my math is nothing. Okay, so that's the first step. Okay, yes. Yes, okay, so that's a good question. So genotype sniffingness, genotype missingness in this case is the genotype call rate, which is what Plink uses to understand SNP missingness, if that makes sense. Remember in computer coding, what's the word? Not syntax. The language that you use is really, really important. And so you have to understand what language the software uses in order to use the software properly. Okay, that's why it's really, really, really important before you want to jump head straight into using a new software. Read through the documentation. Okay, you can see from Plink where you downloaded it, you went to a website where it had downloaded files, but then it also had a whole lot of stuff on the other side with a whole lot of like steps and flags. Okay, this is what we call the user documentation. It's really, really important to read through all of the user documentation to understand what language does Plink use? Like, what must I say to make Plink understand me, if that makes sense? Cool, exactly. Cool, so everyone okay with the steps so far? Even people online? I hope so. I think Yolandi's onto it. Okay, cool. So this is what we specified so far. So now I need to say, so I've basically said, Plink, you must use this file format, and you must use this file, and you must filter out any SNPs that have a genotype call rate less than 95%. But now I filtered them out, let's say, it can't just like filter and then be like, okay, cool, I'm done. I want to write this filtered file or I want to write the filtered data to a new file. Okay, if this is really, really important, so we must make sure that Plink makes a new bed file, and we're just going to have to call it something. So you must tell Plink, okay, you filter the data, now make this data into a new bed file, and we're going to call it reference geno, for example. Okay, it's really, really important to do this because otherwise Plink is going to filter it out, but it's not going to write the data to a file. So you're going to have filtered, but you're going to end up with the same file as you started off with. Okay, makes sense. So this is all just about learning how the software talks and what language must you use to talk to the software. Okay, so you can see I've written out my little command and it must be in this syntax. Okay, it's going to throw errors if you don't use the right syntax. Okay, and then what do? Now I just press enter and you can see how quick was that? Okay, that was like this. Right, okay, so super, super quick. And then now it's given you all the information of what it just did. So it used Plink. It gave options in effect. So it used the B file reference. It put the genotype quality call threshold on. It made a new file and it called this file this. Okay, and then it gave you options or it gave you some information about like how much RAM it used and all that. That's not really important right now. You can see it has 81 samples. We don't have any sex information for these samples because I was lazy and I didn't want to code that. So but we know we have 81 samples. And then we have 389,757, the numbers. Okay, we have this many variants in our BIM file. This is from the original file, right? And then you can see calculating allele frequencies. So it did that. And then it applied the flag and it said, so many variants were removed due to missing genotype data. So now you have 388,641 variants remaining after the filters. Okay, someone said, what is the reference.bim file? We can actually have a look at what that is. So if I want to have a look at what a file is, let's say I just want to look at the first 10 lines of a file, I can just say head. So you can see the BIM file has all the information on every single SNP that is in your data set. So you can see the first column specifies the chromosome. Second one is the RSID. Third column, I think, is centimorgan information. I'm not entirely, I can't remember off the top of my head. It is, okay, good. Fourth column is the chromosome position that it is. So it's in chromosome 1 and it's at this base pair. Then the first column is I think it's the reference first and then the alternate allele first. Yes. Okay, so you can see our reference allele is A, or our major allele is A, and our minor allele is G at the first site over here. Cool, so that's in our BIM file. Happy with that. Okie dokie. Cool, so that's our first step. So we filtered out genotype missingness. Yay, cool, awesome stuff. So now, what is the next step in our quality control? Okay, our next step is we want to filter out, so we filtered out SNPs with missingness. Now we want to filter out individuals with missingness, okay? And we are going to say people who are missing genotypes, people who are missing more than 10% of the total genotypes, we don't want. Okay, we want people who have 90% of the information in the total set, basically. So again, we have to do the same thing. So I want to say, I want to use Plink. Again, you must say which file format you're using. You have to do this every single time you run Plink, sorry. And then which file we're using. I want to use the file that I worked with, or I just made, right? I want to use the genotype file. If you're just going to use the reference file, you're just going to be starting from square one again and again and again. Okay, now we want to filter out missing information in individuals, right? So the flag for that is missing mind. Okay, so mind. Okay, and again, we want to remove individuals that are missing more than 10% of the genotypes. So we want to have a threshold of 0.1. Okay, all good so far. Okay, again, before we click enter, we really want to say, okay, Plink, make me the new file and call it something new. Okay, so let's say genome mind. And you can see, again, ran super, super fast. Okay, you can see that there weren't any samples removed. Yes, Crystal. Thank you. Okay. Hello? Thank you for the heads up. Okay, cool. Everyone saw the command line, right, though? Can you zoom out a little bit? There we go. Okay, cool. So it's run, again, super, super fast. That is awesome. And you can see no samples were removed due to missing genotype data. So this is an important thing to do. You must make sure you put the geno filter first, and then you do your mind filter. Okay. Otherwise, if you do vice versa, you're going to remove individuals, which is a bit of a pity. Okay, I would rather remove a couple thousand SNPs than remove a whole individual with like 1 million SNPs in them. Makes sense. Okay, cool. So that is the mind filter. And again, you can see it wrote all of my output files and it worked so fast. Okay, look at that, a whole 1 second. So it started at 10:22:11 and it finished at 10:22:12. Yes, Caitlin. Okay, I think, okay, perfect. Caitlin is my PhD supervisor, so she's going to tell me if I'm wrong. So basically why you would have genotype data missingness. So this is going back to, remember when you got the DNA in your lab, you put it on that array. And remember the arrays use little probes, what we call. So it's kind of like the little, sorry, people online, I'm using my hands, but they are little probes that the DNA sticks to so that you can get the genotype information. Okay. If you don't have a lot of DNA, or you have DNA that's not of great quality, or even sometimes this just happens spontaneously, sometimes the DNA is going to stick to those little probes. Okay, it's a bit of a hit and miss thing, as we all know in the lab. So not all of the DNA is going to stick to the probes. So let's say if I've got 5 probes over here, not all of my DNA sticks. Let's say it only sticks to like 4. So that you can see I have 1 genotype missing. Okay, so that is where you get genotype missingness. And then individual missingness is when this happens, but on an individual scale. So that one sample doesn't have all of the genotypes because of that array hybridization error. Is that correct? Thank you. Okay. Yes, boss. Okay, great. Cool. So that makes sense. This is basically filtering out any errors we had during genotyping, not errors, but any technical problems that we had during genotyping in the lab. We want to remove them. Okay. Happy days. Everyone all good on this case? Great. Yes. Sorry, I think you understand the question better than I do. You could tell I'm a lab, but I mean, I'm not a lab person. Yes.
That's the chance of other allele basically zero, basically the thousands. So if you want to actually have that mindable alternative allele, much as the alternative, because that's very hard to act as well. And also some of it is a little bit. But if you take into association analysis, actually just introduce some noise with your signals. So if you do association testing, you want to see which one is the strongest, obviously, association, and just introducing that in one person kind of message the strength of the association.
Okay, for the people online who weren't able to hear that, Yolandi was just talking about some technical things with multi-allelic sites. If you want more information, talk to Yolandi Swart. Thank you. Okay. Yes.
Section from Michael. He asked, what sort of inference limitations does relaxing the QC threshold?
What sort of inference limitations? What did you comment? What is the answer?
So you want to actually do as much information steps as possible when you actually do answers. So you want to make sure with the threshold you're excluding any steps that might be false positive or just G2 using differentiate rate. And so relaxing will include those steps. So you're actually increasing your chance of false positive downstream, and also you're actually making that your answer stream is likely wrong based on the spectrum.
Perfect. Okay. Person online, I'm going to summarize that for you very, very quickly. So basically, relaxing your threshold will ensure that you are adding more SNPs, but you're going to be including SNPs that might be technical artifacts or might be technically wrong or maybe a genotyping error. So relaxing the threshold will include more SNPs, but it's might be that these SNPs are wrong. Whereas comparatively, being too stringent with the threshold, you're going to be excluding a lot of SNPs that are wrong, but you may also be excluding SNPs that are true. Okay, so it's a very fine line that you're walking through. I would rather exclude the ones that, yeah, actually, let me stop there. I'm going to stop talking right now before I say something. Okay, cool. So that's just the balance that we play as bioinformaticians. Yes, that sounds excellent. Thank you. Okay, perfect. Everyone online, we are going to be taking a tea break for half an hour. We will be back at 11, continuing the demonstration. I'll leave my thing on shared so that you guys can take notes if you need. Alrighty, welcome everyone back from our tea break. Okay, so we are going to pick up where we left off on our demonstration. So just a reminder about what we did before the tea break. We had a look at how to call Plink, so how to tell the command line, like I want to use Plink. We had a brief look at like what the kind of language that Plink uses. So we remember we must specify each time that we want to use a specific file format, that we want to use which file, and then we started with genotype call, race quality control, and then had a look at individual quality control. So we've already filtered out SNP missingness and sample missingness. Hopefully technical errors have been resolved during the break, and if not, we will definitely do it before you implement your own work. Okay, cool. So the next step, so going back to our lecture, the first step was filtering out SNP missingness. The second step was filtering out sample missingness. The third step, if you guys remember, was filtering out monomorphic SNPs. Okay, and remember, I told you guys that there's a very important equation that you guys must learn, which is the minor allele frequency of monomorphic SNPs in your data set. Okay, so for those who can't remember, that formula is 1 divided by 2 times the number of samples. Okay, so we have 81 samples in our data set. So putting this number into our calculation, who can tell me what the minor allele frequency threshold we want to implement is to remove monomorphic SNPs? Or who can tell me how to calculate it? Yes, excellent, good. So 1 divided by 162. 162 is 81 samples times by 2. The reason why we times it by 2 is because each individual has 2 alleles at every position, right? They've got 1 from the mom, 1 from the dad. Okay, so each individual essentially has 2 alleles. We've got 81 samples. So essentially we have 162 versions of each allele. Well, we have 162 alleles for every individual. Okay, never mind, I'm confusing people now. We have 162 alleles, okay. And then 1 divided by 162, that is 0,006. Okay, and that is our minor allele frequency threshold. Does that make sense before I move on? Cool. People online, I trust that you're just going to pop a question in the chat if you are stuck. Okay, so we're going to implement that into Plink now. So again, the same thing. I want to say, hey, computer, I want to use Plink. I need to use this file format. I need to use the binary files. And remember, we want to use the file that we just had worked on. So that is the geno-mind file. That's the output file from the previous command we just ran. So we want to take that output. That's going to be our next input. Okay. Then we want to filter out, sorry, this mic is annoying me. Okay, then we want to filter out SNPs with a minor allele frequency less than 0,006. So we say, okay, I'm filtering for minor allele frequency now. That's the MAF flag in Plink. And then it's 0,006. That's the threshold that we just calculated. Okay, everything else is the same. We want to say, okay, Plink, you're going to filter that out. Make me an output file and call it something else. Okay, mind, let's just say I'm at the NAF, or I like to say no mono SNPs. Okay, because sometimes you may want to add a minor allele frequency filter for something else. So I like to just, you can actually name these files whatever you want. There's no rule. Okay. Okay, so that is our command. I'm going to press enter. And again, it ran super, super fast. it removed, you can see, so there were 3,000 variants removed due to the allele frequency threshold. Okay, and this is how many variants we have remaining after the main filters. Cool. So Flink is awesome. So it identifies exactly which SNPs deviate from this threshold and removes them for us. Cool. Okie dokie. Happy with that so far. All good. Okay. Awesome, awesome, awesome stuff. So the last thing, well, not the last thing. So the next step in our quality control filter. So we've done SNP missingness, individual missingness. We removed our monomorphic SNPs. So what's the next step? Rare variants, if yes, actually that's fair, yeah. So you can apply another MAF filter if you want to. For our purposes, we're not going to. We're happy with including all the variants we've got now. So the very next step after that is Hardy-Weinberg. Okay, so again, we want to, the exact same thing. We're going to call Plink. Going to be like, hi, Plink. Yes, yes, yes. Ah. Okay, cool. I actually think there's a cool function on here where I can. Evan was playing with this the other day. Ah, okay, there's a pin here. Okay, cool. So how I got that filter, can the people online see this? You can't see that online. Okay, cool, cool, cool. I can annotate. Okay, here we go. Cool. Okay, we're going to try out my drawings, guys. Okay, cool. So where did I get that number from for minor allele? No, come on, annotate. Hello, there we go. Okay. I want to draw, but I don't know how to draw. Okay, I'm just going to try and explain, and hopefully this makes sense. Otherwise, maybe someone can type it in the chat as well. So the formula for calculating the minor allele frequency of your monomorphic SNPs is 1 divided by number of samples times by 2. So if you have 81 samples, we take 81 times that by 2, which is 162. Yeah, 162. And then we take that number and divide it by 1. Perfect. Sorry, 1 divided by that number. Thank you, Crystal, for helping me. Okay, sorry, I couldn't draw. So hopefully, Debbie, that makes sense. If it does, can you just give a little thumbs up to Crystal's answer? Okay, and 1 over 162 is equal to 0,00617, blah, 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 blah. I'm just going to round it up to 0,006. Okay. Cool. Maybe if you can just indicate that that makes sense. It'll be fine, right? Okay, cool. Okay, so we've done our monomorphic SNPs. Where was I? Oh yeah, the next step is Hardy-Weinberg equilibrium. Okay, we want to identify which SNPs deviate from our Hardy-Weinberg equilibrium threshold, and we want to remove them. So again, same thing. So we are going to say, hi, Plink. You're going to take this file, and remember, it's the output from the previous command. Okay, so we always go back to the output from the previous command, the geno, the mind, and the no monomorphic SNPs. The flag for Hardy-Weinberg and Plink is --HWE. Okay, and we, as I think someone asked a question in the last lecture, which threshold do we use? We use 0.00001. If you want to, and if you think it's necessary, you can make it more stringent or less stringent, but this is what we're just going to use for today. Okay, and then again, we have to tell Plink, make my file, output my file, and then we're just going to call it something else. Mind, no monomorphic SNPs. Someone says online they can't see the code. Other people on Teams, can you see the code? And you can see exactly where I am, right?
Yeah.
Okay, I'm afraid that's just you. Maybe just like log off and then log back on again. I'm sorry. Yeah, sorry. If it's too small, yeah, zoom in on your Teams because I'm afraid I can't make it bigger from where I am. It's right at the bottom of the screen. That's correct. If we want to, I can actually, let me actually, oh, Yeah, let me just clear it and start again. Okay, wait. Okay, so I'm starting at the top of the screen again. So, oh, okay. So again, we're just going to call plink2 B file. Remember, we're calling the last file that I was working with. So that's the last file that we outputted. We're filtering for Hardy-Weinberg. We're using 0.00001 as our threshold. And remember, we got to tell plink, make the bed file, and call it something else. Hardy-Weinberg equilibrium. Okay, so this is the command. Again, just press enter. You can see it gives us our little file again. 81 samples. We have so many variants in the initial file, and the Hardy-Weinberg filter removed 900 variants. Okay, and then that gives you how many variants are after the filters. Okay, and then outputs our files. So that's awesome. And those are all the QC procedures we are going to do today. The last thing I want to show you guys is that you can choose to do one step at a time and look at your output files. What's also cool about Plink is that it does output what we call log files. So it kind of documents everything that we have done in the previous one. So let's say this is the last test we just did, right? We filtered for Hardy-Weinberg equilibrium. If you want to, if let's say you did this 5 years ago, a long time ago, and you can't remember what you did or what this threshold is, or oh, yes, sorry. There we go. Okay. If you can't remember what the threshold is, yes. My mind is just blown. Oh my God, you learn new things every day, guys. Okay. Okay, look at that. What? Even me, I learn things. Okay, can you guys see that better now? Okay, cool. So let's say, so Plink also outputs these things called log files, which basically documents all this prints over here, everything that Plink basically just did. It basically prints this into a log file. So you can say cat and then the log file, just to print out what the log file actually looks like. And you can see it's exactly the same as what happened. Okay, so what it printed to the screen, it prints into this log file as well, so that when you come back a couple months later and you can't remember what you did or how many variants were removed or however, you can just open up the log file, take a look at it, and just refresh your memory. Okay, and then the last thing I want to teach you guys is, before you try this on your own, is that you can do this one step at a time, as we just did, or if you are stressed, not even stressed for time, when you become a little bit more comfortable with command line and with Plink, you can actually just do this one shot. Okay, so let's say we're working with the reference. We're going to now, in one line of code, do everything that we just did. Okay, you guys don't have to do this, but this is definitely, when you get more comfortable with this, you can't hear anything. Okay, is this better? This will just have to annoy me. Okay, cool. So in one line of code, we are going to put everything. This is obviously when you are getting a bit more comfortable with Plink and you know what is happening and you understand exactly what all the steps are, then you can start doing this. And it just makes your life a bit easier. Oh, that's not. Okay, so we're applying the same geno threshold, the same mine threshold, the same minor allele frequency threshold. I'm just going to double check. Yes, the same minor allele frequency threshold and the same Hardy-Weinberg threshold. But still, you must say make bed. Still have to say out. And then let's just call it reference QC, because I've put all my QC steps in one go. Press enter, and you can see it did everything for me. One shot, no problems. Okay, so it started with so many samples, started with so many variants. You can see that the geno filter removed so many variants, 1,000 and something. Hardy-Weinberg removed this much. And you'll notice that these numbers, if you remember, you can even check in the log files. These are actually the same numbers as if we were to do them one at a time. So it didn't actually affect the calculations. It didn't affect what was removed or whatever. It just is a bit more convenient for us right now. And then the minor allele frequency filter, again, the same thing. Okay, and that's it. The quality control finished. Yes. Oh, great. Yes. So for this minor allele frequency, if we remember back, this is not your generic minor allele frequency filter. This is filtering out what we call monomorphic SNPs, which are SNPs where the minor allele is not present in the data set at all. So this is only for the major, when the major allele is present. And this will depend on just how many samples you have. So if you have 100 samples, you're going 200, and if they are all the minor allele or all the major allele, then the minor allele frequency for monomorphic SNPs will be 1 over, how many individuals did I say? 100. So it'll be 1 over 200. Okay, so the reason why we can't just use a flat rate is because it depends on how, the calculation depends on how many individuals there are in our data. Yes, yes, exactly. You can do a second filtering step if you wish to remove SNPs with a specific minor allele frequency threshold. As we remember back in our slideshow, which I'm sure Devin will make available to you at the end of the day, it details all the steps. The minor allele frequency, the monomorphic SNP step first comes first before a generic minor allele frequency filter. Okay, for monomorphic, understood. Cool, yay, Crystal. That's a good question. I actually don't think you can. I think you have to do one at a time. That's actually a good question. Yeah. So I think in this case, if you wanted to do just one shot, you can't really say, Plink, do this minor allele frequency and do this minor allele frequency. Remember, because we put in the threshold, Plink just identifies what it is. So Plink doesn't know if you're filtering out monomorphic SNPs or doing a general frequency filter, if that makes sense. Exactly. Exactly, yeah. Depending on your threshold, it would have been filtered anyway. So if your minor allele frequency is 0.01, you don't actually have to do this because it's going to remove them anyway. So it depends on what your threshold is, yeah. Exactly, yeah. Cool, makes sense. Yay, any other questions? Yes.
If you're working with, example, RNA single-stranded,
What?
like 1 over
1, yeah, 1 over n, theoretically. Yes, that's a good question. So if you're working with something like, so remember DNA, oh wait, RNA. So the reason why we multiply it by 2 is because each individual inherits 2 sets of DNA, right? One from their mom, one from their dad. RNA is different because it depends on how many copies you have in a cell at any point in time, depending on what's being transcribed. Oh, an RNA, yes, sorry, yes. So if you're working with an organism that theoretically has only RNA and it only has 1 copy in its little nucleus, then yeah, it would be 1 over n instead of 1 over 2n. Okay, but we're working with humans today, so it'll be 2n. Thank you for, yeah, that's actually a good question. Okay, cool. Last question. So if I understand, you apply like one step and then you want to move on to the next. So it will take it into account, remember, but we must specify the output file. So let's say we make one output file, like let's say we've done the genotype call, and let's say we've applied the genofag, and the next thing we want to do is remove individuals with missingness. We must take this output and use it as this input, if that makes sense. So we must take this file and use it. Otherwise, we're just going to be working with the same file from start. Yeah, the same thing. Yeah, so you're going to have to take the output file again and just Alrighty. So now we've done the reference. So now you guys are going, now it's the implementation, right? Yeah. So now I've shown you guys what to do. Now you guys have to give it a try. Okay. So pull out your laptops and open up your command. Devin should have made the data available for you on that OneDrive. The data is there, right? Okay. So make sure that you download those files and put them in a little folder where you're going to work. And please make sure that you need to have the bed, the BIM, and the FAM file for both sets. So you're going to have a reference BIM, a reference bed, a reference FAM, and you need a target bed, a target BIM, and a target FAM. Okay, you need to have all three. Otherwise, Plink is going to complain. Cool. Yes, so the goal at the end of this, you are going to implement exactly what we have done over here in our demonstration. So the goal is to have applied the genotype filter, the missing individual filter, the removing of monomorphic SNPs, and apply the Hardy-Weinberg filter. So you just have to do those those four today. Devin, it says someone can't access the files. Okay, but you're going to produce those, apply those four filters to both your reference data and your target data. So you're going to have essentially your target and your reference files QC'd at the end of this. Yes. Okay. Always double check how many individuals you have in the file. So check how many individuals you have in your target file and how many you have in your reference file, because your minor allele frequency filter will change for monomorphic SNPs. Okay. Yes. I have something even better. Okay. I knew there was a reason that Devin, I still can't share. Okay. Devin, I still can't add slides. Can I? Okay. Okay, cool. What I'm going to send you guys the slideshow, and I'm going to send you guys some notes so that you guys can keep track of what to do. Yes. Perfect. Okay, guys, so the people online, we're going to stay online. We're going to monitor the Q&A session and the chat. So if you have any problems, please don't hesitate to reach out. And then we will be back again for so you're going to work on Plink genotype quality control in your own time until 12 o'clock. And 12:45, we're back for the next demonstration on merging. Cool. Thank you all. So welcome back, everyone. We have just started now. I think. Can you hear me on Teams? Just give a little thumbs up if you can. Awesome. Okay, cool. Yay. Okay, cool. You guys can hear now. Perfect. Okay, cool. Okay, awesome stuff. So we are back from lunch. I hope everyone was able to work through all the issues that they had with Plink and that we are able to move on. So hopefully you've worked through the implementation of quality control. So you should have, if we see here, you should have your QC reference files and QC target files. Okay, have we all got that? Everyone has hopefully managed up until this part. If you haven't, then you're going to have to do that bit. So you need to do genotype quality control before you attempt the merge. Okay, so what we're going to do now is we're going to demonstrate the merging process. And then we're going to give you guys opportunity to try the merge yourself. But please make sure you finish quality control before you merge. Okay, cool. So now we are going to work on merging. Okay, so excuse me for a moment while I quickly take out my notes. Just for 1 moment. Okay, so you'll see, I think earlier I did put up on the Teams chat like a step-by-steps on how to do your quality control, which I'll also paste this here after the demonstration. So remember in merging, we have to merge our intersecting SNPs, right, or our common SNPs. We can't merge SNPs that are not common because we're going to then end up with missing data, which is going to throw our genotype call rate out of proportion again. Okay, so what we need to do is we need to, first of all, identify which are our intersecting SNPs. Okay, so the first thing we need to do actually is you're going to have a look at, if we were to look at what the reference BIM file looks like. Remember, the BIM file is the file with all the variant information. You can see we have our RSIDs. Okay, so what we're going to do to help us identify which SNPs are intersecting is we are going to just change these RSIDs to a bit of an easier format to understand. We're going to change it to a chromosome-based pair position format. Okay, just so it's easier for the next step. What we need to do is convert all the SNP positions to chromosome base pair format. Okay, all okay with me so far? Okay, so that's just the first step. This is just to make the next step a little bit easier. So we're going to need link 2. Again, we must tell the program what file format to use, and we must tell it which file to work with. So we want to work with the file that we've just quality controlled because we spend so much time doing that. Okay, so that is what we want to use. We're going to say, we're going to set all the variant IDs. So this second column over here, this RSID column over here, we want to change this from RSIDs to chromosome-based pair position. So Plink understands this as an @ hashtag symbol. Okay, make sure you put the little quotes around it because that's important for your computer to understand it and read it. Okay, so once we have told Plink, we don't want RSIDs, we want this format of our IDs. So we set it to change it. We again make our, we say out, and then we're just going to call it something else. So reference QC, and then I'm going to say this is a file that has chromosome base pair. Okay, run that. And now if we have a look at what this file looks like, what the BIM file looks like, can you see the difference? So you can see the second column, instead of having RSIDs, we now have chromosome base pair. All good so far. Okay, so this is just a bit of a technical step, but this is just really to get the format right. Okay, so we've done that for the reference file. Just need to do that for the target file. Okay, so I'm going to be a little, so we can have the exact same command, but this time, instead of using our reference file, we want to use our target file. Okay, but everything else is the same. We're calling Plink. We're saying, hey, Plink, take a look at this B file. Set the IDs to the right formats I want. Make a new file, and then it will do that for us. Okay, so that's just a little bit of a technical step, but are we all okay here? We're just kind of reformatting the files a little bit. Okay. The next thing we want to do is we want to make a list of all of the variants in these files so that we can identify what are the common variants. Okay. So to do that, we're going to use a command called awk. Okay, which is basically just telling us to, awk is kind of like a looking command. So it's going to say like, look within this file. Okay, so we're saying awk. Okay, this is going to be a little bit of a technical coding thing now. So keep in mind, but I am going to give you guys the codes when the demonstration ends. Okay, so we're going to say awk. So look in this file, print the second column for us. And then which file are we looking at? We're looking at the reference QC files with chromosome base pair position. This time you must end it with them, okay, because we're looking at that file specifically. We're looking at the variant file. Okay. For the next step, we need to sort this file. So we're just going to do that all in one line because it's a cute little line of code. And then we're going to just call this, put this in a new file. So we're going to call it, I just like to call them SNP lists. Okay, so we have said, okay, this is the reference file. Well, this is the variance file. we want to get the second column of this file that has the chromosome and the base pair position. We need to sort it just for the next file. This is just going to sort it into the right number format for the computer to understand. And we're going to print it out into a new list. Okay, so that's going to run. And if you have a look at what this file looks like, sorry, there's a lot of files there. Can you see it starts at chromosome 10, and then it gives chromosome 10 ... our SNP position. Okay, the reason why it starts at chromosome 10 is because we've sorted the file. So Linux likes to sort things 1, 0, whatever, and then 1, and then 11 there. So it's just the way that the computer does it. That's really important to include that step. Okay. Okay, all good so far. Cool. This is just the technical parts. This is just the little nitty-gritties of getting the files into a good format so that we can move on to the merge. Okay, so we've done this with the reference file. We need to do the exact same thing with our target file. Okay, so again, target QC. We want the chromosome base pair one, and we want the BIM file. We want to sort this file into the right format for the computer to understand. And then we're going to put it out into a list. Okay. So we've looked at that. Let's have a look-see and make sure that things are looking great. And you can see I have a file also with chromosomes, SNP positions, and you can already see, I'm sure, that in the reference file, we have this SNP, and then in a target file, we have this SNP. So you can see that's already one SNP that's common between the two files, right? And that's going to be one of the SNPs that we merge. But this file has got like 300,000 SNPs. I'm not going to go through myself and find which ones are common, right? So that's where we come to our next line of code, com, which is basically saying, look for the common lines between these two files. File 1 is my reference SNP file, and file 2, is my target SNP file. So I want you to look for the common SNPs or the common lines between these two files. And then let's put it out into a new file so that we can have a look at the intersecting SNPs. Okay, all good. Everyone okay? So this is all the technical stuff, but this is just line by lines of code that we just need to run in order to find what we need. Okay, and I am going to make these available to you, but as long as you understand what the codes mean. So you're just trying to identify the columns that we need, we're going to sort them out, and then we're going to identify what's common between the two. And then you can have a look-see. Now I've got a little file called intersecting SNPs. And these are all the SNPs that are intersecting or which are common between the two files. Okay, so remember our little Venn diagram? These are the SNPs that are in the middle. Okay, cool. Okay, this is I did know this. This I will double check, but I think it means in the first, what's the word? Like the first instance and the second instance. So the dot dot is, I think, the separator between the two like instances. I'm going to call them that. And so it's looking at if you want both the first and the second column. So we want to look at both the chromosome and the SNP. Okay, because remember the SNPs are just showing where on the chromosome it is. So if we just look at the SNP position, there's going to be a couple hundred of this number. Okay, so we want to look at both. We want to include basically both the chromosome and the SNP position. I hope that makes sense. Sorry, that was a lot of my rambling. Okay, Isabel, you can just give a thumbs up if you understand. Otherwise, I'll type it nicely in the chat now. Okie dokie, cool. So basically, it's basically just making sure that we include both the chromosome and the SNP position. That we want to look at both of them together. Okay, there's just like a coordinate. Okay, enough of my talking. Okay, cool. So now we have a file of all the SNPs that we need, right? Okay, so now we need to make sure that we've identified these common SNPs. what's the next step if we go back to the lecture that I gave this morning? Is that we need to extract these SNPs from our files. Okay, so that's what we're going to do. So we're going to use our best friend again, Plink. Okay, so back to Plink. We're going to say, okay, first we're going to work with the reference file. The one that we have spent so much time QCing, we're going to use that one. And make sure we use the one with the chromosome base pair position. Okay, so we want to use the reference file, the one that we QC'd, and the one that has now the correct file format. So the chromosome base pair one, not the RSID one. Okay, it will throw an error if you do use the wrong file, and then you can just go fix it. Okay, then we want to, so we have this list of SNPs. We went
testing 1, 2, testing 1, 2.
What a time. Okay, so we have this list of SNPs. We want to extract these SNPs from our reference file. So we use the flag -- extract the intersecting SNPs.
Testing 1, 2. Testing 1, 2. The PA system is just being tested. I repeat, the PA system is just being tested.
Okay, thank you. Okay, so we're going to extract these from our files. And then the same thing as we've done with all of our Plink commands, we're going to write a new file. So we have to say make bed out. And then I'm going to call these the reference, the common SNPs. Okay.
Testing 1, 2. Testing 1, 2.
Okay, so you can see we started with so many variants in our QC file, and now in we've extracted all the common variants. So we have a little under 300,000 common variants. Okay, so we've removed a lot. That's just because there were so many variants that weren't found in the target file. Okay. Okay. Alrighty, sorry everyone. Okay, cool. So now we've done that for the reference file.
The PA system is just being tested.
Okay, so we've done this with the reference file. Now we need to do the exact same thing with the target file. Okay, so again, this is very repetitive, but it gets the job done. Okay, so again, we're calling on Plink. We're working with our target quality controlled file, the one with the correct format, so the chromosome base pair position one. Okay, again, we want those intersecting SNPs, so we're going to say extract the intersecting SNPs, and we're going to make it into a new file. This time I'm going to call it target. Okay, and you'll see here that we have the same number of SNPs in the reference file as we do in our target file. So the SNPs are now overlapping. We shouldn't have generated any missing data. We shouldn't have missing data when we merge the files together. Cool. All good so far. Everything making sense. Excellent. Okay, so now we come to the fun part. Now we can finally merge. So just so you know, I'm sorry this wasn't in the email when you guys registered, but Plink 2 doesn't merge, Plink 1 merges for some strange reason. Okay, so we're going to need, instead of using, you can see my previous commands all used Plink 2. Now I'm just going to use Plink 1. Okay, but you have to make sure you download both. Okay, so that's just another thing to deal with when we are in the implementation phase. Okay, so this time I'm going to call on Plink, normal Plink. And now we get to merge. Okay, so the first thing is when we are merging with Plink, we need to specify the first file that we want to merge, which can be either reference or target. I'm just going to call it the target file. So target the common SNPs. Remember, we can only merge the common SNPs together. So we need to merge this file. Okay. Okay, so now bear with me on this one. This is going, so now, but for the next thing, when we want to merge, we have to specify the bed, the BIM, and FAM file in that order for the next one. So we're going to say B merge. So we're merging the binary file. So we're telling Plink, this is my starting file, the target file. Merge it with these binary files, which are my reference. Common SNPs, the bed file, the reference common SNPs, the BIM file, and the reference common SNPs FAM file. Okay, you must specify all three, and you must specify them in that order, BED, BIM, and FAM. Otherwise, Plink's going to not know what you're doing. Okay, so we have to say, Plink, this is my starting file. Merge with these three binary files, the reference, BED, BIM, and FAM. Then you're going to take this merged file, make a new file. So make BED out, and let's just call it the merged files. Okay, ready? This is always the point where you look at your code, you take a deep breath because you don't know if you're going to get errors, and you press enter. And look at that. Did it merge? Yes, it merged. Okay, great. Cool. Okay, cool. You always have to, okay, cool. So you can see, so I ran that quickly. Okay. So you have this many markers from your target file. This many markers, remember markers are SNPs, from your reference file. So this many are present in the base data set. The base data set is the target one. That's the first thing that you specified, the first file that you specified. So performing a single pass merge, I don't know why it's, yeah, actually I do, sorry. It's performing a single pass merge because it's small enough. If the data set was much bigger, it'd have to do the merge in like several steps. But you wouldn't have to specify that, it just do it on its own. And then it merged the files created. So you can see in our target file, we had 14 people. In our reference file, we had 81 people. So we've merged them together, we have 95 people. So then math is mathing, we're happy. No one got lost in the process. Yes, and you can see our genotyping rate is 99% basically. So we don't have a lot of missing data, which is so. And that's it. That's the merge. So it's done. Okay, so we've merged it. And now as a sanity check, remember in the lecture, I think someone asked a question, if you have to quality control after you merge. I do it just as a sanity check and also just to make sure that nothing has gone funky. So we can do that again. And you guys are pros at quality control. So I don't have to talk you through this, but we're just going to, again, use Plink. E-file. If I want to have a look at the merged file, again, I can use the same thresholds. So like at my Geno 0.05, my Mind 0.1, my Hardy-Weinberg. I'm not going to the monomorphic SNP filter because I think we dealt with that earlier. And generally speaking, you don't need to do, there's a lot of echo. I'm sorry. Is it okay? There was someone testing the intercom earlier. Okay, but we don't need to worry about the monomorphic SNPs at the moment. Generally, you can just apply a Geno filter, a Mind filter, and your Hardy-Weinberg filter. Again, just say make bed, out, merged files, QC, run that. And so you can see, so 0 variance removed due to missing genotype data, which makes sense. We saw after merging, our genotype call rate was 99%. So none of our, like, on average, our SNPs don't fail the 95% threshold. 161 variants were removed due to Hardy-Weinberg's. Cool. No people were removed. Yeah, so our file is merged, yes. Cool. Yes. Okay. Yes, that's true. Exactly. Okay. Yeah, that's actually a great point. So seeing as we have a bit of time, we will show you why we take such time to make sure we're merging the intersecting SNPs. Because if I were to do just summa plink B file, let's say I were just to take my target QC file. So this is the one before, like we finished right at the end of our first session, right? So this is before we did any of the intersecting SNP stuff. This is exactly why. So So we're taking that file, we're just doing B merge. So this is what not to do. Okay. We've got our reference QC.bed reference. You can see the commands are all the same though. Reference.bim, reference.qc.fam, fam, make bed, out, and then what do I call it? Wrong merge. So do not do this. Okay. So you can see my genotyping call rate, it's not terrible, right? It's 96%, which is fine. But that means that when we apply our genotypes filter, we're going to have quite a few SNPs removed. And so we can do that now. So remember in our previous, in our previous quality control, you saw no variants were removed when we took the time to make sure we were merging the intersecting SNPs. Whereas if we now don't take that time to merge the intersecting SNPs and rather do the wrong merge, this, and we just apply genofilter, okay, look at how many variants we had to remove. 98,000 variants. In a file of just like 200,000 variants, that's a lot. Okay, cool. And this becomes so much more complicated when you have, we're just merging 2 data sets now, but imagine you want to merge like 6, 7, 8 data sets. It does happen. So then the more missing data you introduce by adding in all of these extra SNPs, the lower and lower your genotype call rate is going to be. And eventually you're going to end up taking lots of SNPs out. So this is why we don't do this, okay? And why we rather look at the intersecting SNPs, make sure we remove those, because that will make sure our genotype quality is chef's kiss. Okay, cool. Makes sense. Yes, and that is merging. So you can see it's very, very simple to do, but very important to do right. A lot can go wrong. Okay, if we do not follow our little steps. Okay, we've got some time for questions. So any questions on merging? So just remember this last little bit that I did here, this is what not to do. Okay, do the things before. Cool, but if there are no questions, yes. I will, don't worry, I will. Cool, cool, cool. So like I did with the genotyping, these are just all the steps, especially in the beginning, like the little technical steps with ORC and that kind of stuff, that can be a bit hard to remember. Okay, cool. There is a question. Why do we have a reference and a target file? Okay, so this is like 2 kind of options. Firstly, because it's a nice way to practice merging, just by merging 2 different files. But this is really, really important tomorrow when you are doing admixture with your Landi. So your target individuals are, let's say, the samples that you got for your lab, the samples that you are investigating. And your reference samples are from like 1,000 genomes or whatever. These are populations that you know, or people with population identities that you know. And you have to merge them together if you want to do ancestry inference. So you can kind of infer the ancestries of your unknown or your target samples based on their similarities to your reference samples. Yes, good. And then for admixture, they need to be in one file because admixture only accepts a binary file with both reference and target individuals in one. Cool. Okay, does that make sense? Cool. Well, yes, excellent. Okay, cool. So that makes sense. Great. So we have done merging. So now you guys are going to have until 2 o'clock to do your own merging. So you have your QC files. So you need your target. Oh, wait. That's a nice question from Eugenia. I've heard that merging SNP data from different chips can be tricky. Any comments on that? Yes. So this is very, very important. So this data set is simple to merge because they are essentially genotyped on the same array. So it's from the same chips. So the same variants have been genotyped. Whereas if you're using one array and one array is an H3 Africa array, shall we say, another one is an Illumina array, another one is an Affymetrix array. These arrays will all have different numbers of SNPs, but also different coordinates of SNPs that are genotyped. So different SNPs and different SNP numbers will be genotyped. And that's why it's so important to, okay, so firstly, the one thing that we can do is imputation before we merge. Okay, so the next thing that you're going to learn is imputation after we've merged. But if you're merging data genotyped on different arrays, it's better to impute the missing data before you merge so that you can add in the missing or the ungenotyped SNPs. So you can increase the number of common SNPs among all of your data sets. Okay, so the way to work around that is to impute first and then move on to the merging step. We're kind of doing it backwards now for ease. Yeah. Cool. Okie dokie. So I think that's all from me. Yes, exactly. If you were to merge from different arrays, your genotyping core rates will be so different. I mean, not will be because the different arrays have different SNPs, genotypes. If you were to merge before imputation, you'd have a lot of missing genotypes and missing data. So you'll be removing a lot of individuals and a lot of SNPs. This is annoying me. A lot of SNPs if you were to merge before imputing. So it's better to impute first and then merge if you're working amongst different SNPs. Different chips. Cool. Okay, well, that is all from me then. I will share this code with you to give you the step-by-steps on how to merge our files. And then you guys have until 2 o'clock to try this on your own. And then we are going to have an imputation and phasing lecture and demonstration. Great. Cool. We'll still be here for the online people. We will still be online so that if you have any questions or any difficulties, you can ask your questions.
Okay, let's get started again on the imputation and phase. I hope everyone successfully generated their merged QSEED file, as we're going to take it forward in the demonstration after this very short lecture. If not, post in the chat and we'll see if we can help you out. Okay, so we're going to move on now to the next steps of imputation and phasing. We're going to talk about the basic principles of imputation. We're going to go through a basic workflow. We're going to talk about why imputation is needed. We're going to talk about imputation tools, common imputation tools that we use, as well as the reference panels. We're going to talk about phasing and the basic principles of phasing. Why is phasing needed? Why do we need to rephase before and after imputing? And talk about a few phasing tools as well. Okay, so I thought to introduce the basic concept of imputation. The term imputation can be applied to various fields. It's in a very simply put, it is the filling of missing information. In genomic imputation, it is filling in of unobserved genotypes. So you'll see in the figure on the left, you'll have a study sample that is generally based on array data or genotyping data, and you'll have reference haplotypes or a reference panel that will be without missing information, will be very dense, generally whole genome sequencing data, high coverage, very good quality data, large scale. And you will then compare your study sample, in essence, to your reference haplotypes. In terms of similarity, you will then fill in the missing genotypes. I'm not going to get into the statistical inference behind it, but that is in essence what happens. As I said, it requires a large set of reference haplotypes for accurate inference. The denser the study genotypes, the more accurate imputation. If you can imagine, you are imputing 30 million sites and you only have 200,000 sites in your genotyping file, you can imagine that the chances of you getting low quality imputed variants is a lot more than what it would be if you had 600,000 variants in your genotyping file, or even more. So it is very important to keep that in mind. You also want to make sure that you have the lowest possible FST. Remember, we talked about FST beginning of the day. That is a measure of population differentiation, you want the lowest possible FST possible between your study and reference populations. So if you, a common problem that occurs is you want to impute data based on African populations, and you use European reference haplotypes to do that. That's not going to work. It really is not. So you want to compare apples to apples, in essence. So that is definitely something to keep in mind when you choose a reference panel, but we'll talk about that a bit later. This process of imputation is computationally intensive, very much so. So running it locally is not an option, particularly for large scale genotyping data. Okay, so this is the basic workflow of imputation and phasing. I'm going to run through it very quickly. Let me just get my laser pointer. Okay, so you start with your genotyping data. This will be basically what you've just generated, the files you've just generated. You want to, first things first, is that it is aligned to the reference genome. Generally, it should be, but we always like to realign it anyway. So we're not going to do this ourselves today, but in future, if you ever need to do this, this is a very nifty command to use, but it requires a FASTA file, which is incredibly large. And I think for GRSH 37, it's about 32 gigs worth of data. So we're not going to ask you to download that now, but it's just something to keep in mind. So you always want to make sure that it's aligned. Okay, so you have two options when you do imputation. You can either do imputation using imputation server. So this could be servers like the Sang imputation server, the Michigan imputation server, and TopMed also has an imputation server. And this is basically a website that has a user interface where you upload your data, you do a couple of clicks, choose a couple of options, and you send it to the cloud, and it imputes, and you get a link back with your imputed data. So it's very user-friendly, but there's not much room for what's the word I'm looking for, adjustments and making it specific to your situation. So to do that, you would run it locally or on HPC, high performance computer preferably, because as I said, it's computationally intensive. So in either way, you would need to ensure that you are selecting or using an appropriate reference panel. So we'll talk a bit about the available reference panels now, but you want to ensure, as I said, that the FST between your genotyping data and your reference panel is low. If you're working on the imputation server, you are given the option to select which reference genome your data is aligned to. So generally, as I said, GRSH 37 or 38. You can select your pre-phasing and imputation algorithm, so you can decide what algorithms you want to use. For some of the imputation servers, you're able to do this, and for others, there's defaults. So on the server, they do pre-imputation phasing automatically, imputation, and then you would need to do post-imputation phasing only if you're using the Sanger imputation server, because they stinging and don't give you phase output data. For the others, they do. No matter which option you decide on, whether you do the imputation server or locally on HPC, you also need to do post-imputation QC. So we'll go through all of these steps. For the sake of the workshop and for ease, we won't be doing things locally or on HPC. We will be taking the easy way out and using imputation server. I see there's a question. Do we align SNP? So what the alignment does is it'll align the strands. So it'll make sure that you are looking at the correct alleles when you are comparing the reference and the target data. Otherwise, if you compare forward and reverse strand between the two, then you're going to have an allele mismatch. So it aligns it based on forward strand. Okay. So why is imputation needed? You boost the power of association analyses, you have more data. So you go from 1.2 million SNPs in the genotyping array to 30 million SNPs. So you can imagine the power for association analyses increases. Excuse me. You increase the SNP density for population genetic analyses. So for admixture, for instance, you have a lot more information to play with. For selection scans, you have more of an opportunity to identify variants under selection. And imputation, which is very recent, it can be used to confirm genotypes in low coverage sequencing. So low coverage sequencing is anything below, I would say, less than 8x. So imputation tools that can be used, and this is both on the imputation servers as well as doing it locally. You can use the positional Burrows-Wheeler transform, PBWT for short. Sorry, you can use goodness. You can use Minimac, you can use Beal, and you can use Impute. And each of these have positives and negatives. So you will need to do some research in terms of which is the most suitable for your data set. Okay, imputation servers. So I mentioned a few of them. Allows individuals to upload study genotyping data and perform imputation against the reference panel. It's all automated, very easy to use. So depending on your imputation server that you use, you will be able to select different reference panels that you will use to do the imputation. We have 1,000 genomes. TopMed have their own reference panels. You have the Haplotype Reference Consortium, African Genome Resource, Kappa, and you can have your own reference panel as well. If you have large-scale whole genome sequencing data, that's high coverage. Okay. Can I just ask someone to get me water, please? Sorry. Got a tickle in my throat. Thank you very much. Okay. Phasing. So as I've alluded to, you can do pre-imputation phasing or post-imputation phasing, but we'll get to that now. Let's just discuss what phasing is. So you have a maternal and a paternal chromosome. In routine life, in what you find day to day in your genotyping data is unphased data that is collapsed into a single mixed sequence. You don't know whether it is from the maternal or paternal chromosome, and phasing resolves this. It will tell you from which chromosome it is, and each haplotype is then assembled separately. So phasing is the process of separating a mixed sequence into maternal and paternal haplotypes. So in this process, having related individuals can be very, very beneficial because you in essence know then the phasing, you know what the maternal and paternal haplotypes were, so you can go back and do a sanity check. So trios greatly assist with this process. So as I said, you can do pre or post imputation phasing. Post imputation phasing is only for Sanger imputation server. So you may ask yourself, well, why do you use Sanger imputation server then? Sanger imputation server is houses one of the best reference panels for African populations, the African Genome Resource. If that is the reference panel that we use for our imputation phasing, and it's been shown to be the most accurate. So I would really recommend going that route. But today, for ease, we will be using TopMed. It's equally been shown to be accurate and it's quite a bit more user-friendly. Okay, so why is phasing needed? You obtain a complete picture of genetic variation. You analyze, you are able to analyze compound heterozygotes. You are able to identify variant linkage. And pre-imputation phasing is needed to increase imputation accuracy. So this should always be done before your imputation step, whether you do it locally on the HPC or whether it's built into the imputation server pipeline. Okay, so phasing tools, there are three main ones, although there's probably others out there. ShapeIt, Beagle, and Eagle. ShapeIt has been shown to be the most accurate as far as I'm aware, so I would strongly recommend that, and that is the one that's used by Sanger Imputation Server as well. Okay, so after your imputation, or your pre-imputation phasing and imputation, you want to do post-imputation phasing quality control. The first thing that you want to do is you want to do a minor allele frequency filter to remove non-polymorphic sites. So again, similar to what Dana was mentioning, you can do this to remove monomorphic sites, sorry, non-polymorphic sites or monomorphic sites, or you can do a standard MAF filter. Here you'll see in the command that we use 0.01, so it's completely up to you. We will be using 0.01 for the workshop. You also want to filter on info or R-squared scores. So these, the score is a accuracy score or a performance score that the imputation software provides. So is it that this imputed genotype is what it said it is? And there is no right or wrong answer in terms of the value for this. It's actually quite an interesting field at the moment. People are trying to figure out what the best filter would be depending on your application. So for instance, if you are doing association testing, you want to make sure that your variants that you've imputed are true. So you might want to use an R-squared value of 0.95, but then again, it's going to remove a lot of your variants. So if you want it to be less strict, I would suggest 0.8 or even 0.6. Again, you can start low and go high and see how many variants are removed in each step, and then decide an R-squared value. For today's purposes, or the workshop purposes, we will be using a R-squared value of 0.8. So the commands that you see here are BCF tool commands. Don't worry about them now. I have a separate document for you that will assist you with your post-imputation phasing QC. Okay, so all of that's done. I want to give a brief mention on imputation of and phasing techniques in admixed African populations. We've done quite a bit of work on this, and as well as the recent article, Gupta and Ananyo on the performance and accuracy of various panels and imputation platforms. And as I've said, Sanger Imputation Server with the African Genome Resource is your way to go for imputing African populations, or TopMed with the TopMed reference panel. That would obviously be second best. And your phasing, you would use ShapeIt. So that's just a suggestion going forward if you ever want to do imputation and phasing. Okay, so that is me in terms of the lecture portion. Let me just pull up what I need for the demonstration and we can get going. Okay, so the first thing I'm going to show you is just the different imputation tools that are out there. The first one is the Sanger Imputation Service. So for this, you will, it's very easy. You will put in your name, spelled correctly, Again, spelled correctly, Caitlin. Put in organization, put in your email address. Email address is very important to get right, otherwise you're not going to get the upload and download links. And your Globus user identity. So Globus is a tool that enables fast transfer of genetic data files. To use Sanger Imputation Service, you will require a Globus account in order to facilitate the transfers. And this is one reason why we're not going to use it today, because that setup is quite tedious, but worthwhile. Okay, so you fill in all that information. You then can label your job. Okay. You can select your reference panel. So again, you have the Haplotype Reference Consortium, African Genome Resource, 1,000 genomes, UK 10,000, and then a combination. So for the sake of this, we're going to do the African Genome Resource. You can then select your phasing and imputation algorithm that you want to use. So you can pre-phase with Eagle2 and impute. You can pre-phase with Shaper2 and impute. Phase with Eagle2, no imputation. Impute with PBWT and no pre-phasing. So you have the option of any of them. Once you click submit, you will take you to this page. It will send you an email with an upload link. You upload the data. It does the imputation phasing for you. It sends you another email with the download link, and you download your files. It's that easy. Okay. So for the workshop, we're going to be using TopMed. TopMed is even easier. Okay, you will need an account for TopMed, but I'll give you that information shortly. You put in your password. I see that there's a raised hand, but I see there's a comment. Let me just open my Teams. Okay. If you are using samples from different continents, do you impute with their references from their continents? Can you use, for example, the African for African samples and the other panel for other samples? Yes, that would be my recommendation, definitely. Alternatively, use a reference panel that has both. If you are working with populations with Khoisan ancestry, which is what we routinely do, I would strongly recommend the African Genome Resource. So split your data up into Eurasian populations and African populations with Khoisan ancestry, and then do 2 sets of imputation steps. Yes. Yes, so that's a good point. So the African Genome Resource includes all individuals from 1,000 genomes as well. So it should do a pretty good job with imputing for European populations, but I haven't tested that, so I don't hold me to it. I would recommend TopMed for European populations, so it's just better. Okay, second question, is there a way that imputation can be used in parentage verification where only samples from mother, offsprings, and half-siblings are available? To be honest, I'm not sure. I doubt it. From the top of my head, you would require quite confirmatory results for something like that, and I don't know whether imputation would be able to give you that level of confirmation to do parentage verification. Unfortunately, the webinar does not have the capacity to unmute, but I can try allow mic. Let's just ask your question. Let's see if we can hear you. I've unmuted you, so you should be able to talk. Okay, if you have a further question, just post it in the chat and I will definitely answer it. Okay, TopMed imputation server. So we've logged in and we want to do a run. So we go up to the top, we click on run, we click on genotype imputation. Again, similar to the imputation server, you put in run ID. It can be spelled incorrectly, that's fine. You have a reference panel that you would need to select. And here you would upload your files. So I'm actually not going to show you that now. You'll do that a bit later. Those would be your VCF files that you would upload. You would select what build your data is currently aligned to. You can have an R-squared filter. So if you only want steps outputted that has an R-squared filter or R-squared value of more than 0.3, then you have the capacity to do this, or you can output all of them, no matter what the R-squared value is. You can select whether you want TopMed to phase or not to phase, and they use Eagle for phasing. You can select, again, what reference panel you want. You can select whether you want to do quality control and imputation, or just phasing or quality control only. You need to do some selections and you submit the job and it runs. We'll be doing this now, just now. Okay, so what we want to do is we want to use our merged files. Let me share my desktop screen. It's tidy. Okay, so we have our merged files underscore QC bed bim fam. So what we first need to do is we need to convert these files into the required input format for imputation. The second thing we need to do is we need to split these files so that we have one file per chromosome. Okay, to do this, we are going to use Plink again. And because we want a set of 22 files, in essence, we are going to use a very simple for loop. Okay. So what we are saying to terminal is we are saying the command that I'm putting in now, I want you to do it 22 times. And each time you're going to go to the next chromosome. So you're going to run Plink 22 times. Okay. Yeah. Okay. So I'm migrating to my Plink instance. We are specifying the B file again, which we have merged and QC'd. We need to tell Plink we only want specific chromosomes. So here, as I said, it will iterate over all the chromosomes. So you have dash chromosome, and this $ with the I in the parentheses will be replaced with whatever number chromosome you are doing. So it'll start at 1 and it'll go all the way to 22. Okay. We need to tell Plink that we don't want binary Plink files out. We don't want bed, bim, and fan files. We want a VCF. So we need to tell to recode the Plink files to a VCF. And then we need to specify the output file. Okay, so I'll use input file name. I want one file per chromosome. So we need to change the file name every time. And then we need to do done. Okay, again, I'm going to give you all of these commands. So again, we're using Plink. We're inputting the merged QC files. We're doing it. We are telling Plink to give us only a specific chromosome number at a time, and we're outputting then that chromosome in a form of VCF file. Okay. So now it's going to go really quickly because the files are really small. And you'll see here again, it's splitting it per chromosome. So you'll see the last one is chromosome 22. And it has outputted 95 individuals. It has outputted 4,840 variants that is on chromosome 22. And it gives you a genotyping rate. So if we look at this VCF file, this is what it's going to look like. Okay, again, it's exactly the same information as what is in the BED, BIM, and FAM file. It's just packaged differently. And this is the required format for imputation. Okay, so you'll see now that you will have, amongst the log files and other files, you will have a VCF for each chromosome. That is what you're looking for. Okay. What you now need to do is, because these VCFs are large, in this case it's not large because we have a little individuals and few SNPs. In most cases, your VCF is really large. You can think uploading a really large file to a website is going to take forever. So what you need to do is you need to zip it, in essence. Okay. But we need to use a specific tool to do this, and it's called BGZip. Okay, so here I'm doing a for loop again because we have 22 files that we don't want to do this command 22 times, so we're using a for loop to iterate over all of the files. Okay, so now we need to specify the input file that we want, and simple as that. So it's do BGZip your file that you want to zip, done. Okay, so for this now, it'll zip our files and it'll add a .gz at the end of the VCF, like that. So you'll see there are all of your VCF files. Okay. Now this is what you upload to TopMed. Easy as that. Okay, so let's go to TopMed. Okay, we can give it a proper name. Select files, you migrate to your files, you select all the files that you want, so chromosome 1 to 22. They will all be there. Again, you can you can't impute the Y chromosome, so TopMed or imputation doesn't really occur, doesn't really do very well on the Y chromosome imputation. X chromosome is fine, works pretty well. So if you would want to include the X chromosome in this imputation step, you can do that as well. Okay, so we know that this information is on RCH37. We're not going to do an R-squared filter because we're going to do post-imputation QC. So we are going to filter our data. We can use Eagle for phasing, pre-imputation phasing, the TopMed panel. You select those two conditions and you submit the job. So you'll see that the data is uploading pretty quickly, and that's because we have zipped it. So what TopMed does is, because the TopMed reference panel is based on a different reference genome, GRCH38's newer reference genome, what TopMed will do is it will lift over your coordinates in your file to the new reference genome. So this is done automatically. You don't even know about it. The only thing that you need to know about is that the output file that you get will not be what you gave it, GRCH37, it will be GRCH38. So just something to keep in mind when you use the imputation files. It won't have any impacts on downstream analyses other than if you are merging with other data or if you are looking up a particular SNP. You need to make sure that you know your data is on GRCH38. Okay, so you'll see there's an input validation step. So you have 22 valid VCFs, you have 95 samples, you have 22 chromosomes, 286,000 odd SNPs. It's segmented into chunks for imputation. You can just verify that the data you gave it is in fact unphased, that it was HG19, the reference panel that you selected, and the phasing and the imputation. It says here, as I said, that the uploaded data is HG19 or GRSH7. It's lifting it over to GRSH38 or HG38 because the reference is GRSH38. Okay, so what it'll do now is it'll lift over all the files, and it will output some statistics in terms of mismatches between your reference panel and the data you gave it, mismatches in terms of alleles. So the reference panel says at base pair position 2 it's A and it's T on the target. So that'll come up as a allele mismatch, and it will either, if it can't resolve the error, it will remove the SNP from the imputation step. Okay, so let's leave this to run. What I'm going to do is I am going to post all of these steps in the Teams chat. What I suggest you do first is go onto TopMed's website and create an account for yourself, fill in your information, and then we can come back and look at what TopMed has given us in terms of the quality control information, and we can take it a step further. So I'll post the link to the TopMed website and go and create an account for yourself, and we can come back and carry on. Okay, so everyone should have created an account by now. If you run into any problems, let me know. Our TopMed run is not finished analyzing at the moment. Seems like TopMed server, as it is a bit overloaded at the moment. So what I'm going to do is I'm going to send you the steps and up until uploading to TopMed, run through that, and we can then look at the quality control measures it gives out together. So I'm going to post the steps in the chat. There is a one step where you need to install BGZip, but I've included the command on that. That'll work for both Windows and Linux or Mac. It won't work for that won't be for Mac. It'll be for Linux and for Windows only. If you need the Mac command, just let me know. So work through these steps. I'll give it until 3 o'clock, and we can come back at 3 o'clock, and hopefully everyone would have uploaded their data to TopMed by then. Okay, we'll see you at 3 o'clock again. Okay, so I know that quite a few of you are still waiting on your activation emails. And I can see that TopMed, the server, is incredibly busy. So what I'm going to do is I'm going to show you a screenshot of the previous run we did yesterday. So this was the same input file. So you can see that based on the quality control, which is mainly looking at the compatibility between the reference panel and the your input data, only 128 sites were excluded from the imputation step. So you have remaining of 280,000 odd sites. And this just gives information about how it's divided the SNPs up into chunks, at least 20 SNPs per chunk, and it excludes chunks with more than 20 SNPs. So what will then happen is, so once you receive this information about the quality of the data, you'll see the next step where it shows the pre-phasing and imputation, and you can see that all your chromosomes are shown there. If they're in gray, it's waiting to be imputed, so it's queued on the server. Once it's blue, it means that the imputation is running, and once it is complete, it will show in green. And what will then happen is, so here is the same thing, right? So you go at the top to the results tab. Once imputation is done, these files will be shown here. And as you can see here, imputation results. So you'll have chromosome 1 to chromosome 22. Zipped files, you can see they are really large. So if you click on the top right here, wget, it'll give you the links to download your data. So you can either use these links that are per chromosome. So you'll need to use do this 22 times. So if you just want one link to download all the files, this will be this command here, the curl command. You say copy to clipboard, you open up your terminal, you copy and paste, so you paste, and this will then download all your files from the imputation server for you. Again, as you saw, the files are really large, so it's going to take you a while to download the files. So what I would recommend is that you submit your files to TopMed tonight, or at least this afternoon, and you try to download your files later tonight or at the latest tomorrow morning, so that you have the files ready for tomorrow for the global and local ancestry inference. Okay, so that is your homework for today, to make sure you have those files. Yes, and make sure that RFMix and admixture are installed and ready to use for tomorrow. If you do have any problems with that, please post in the chat and we'll help you. Just as a note, these are zipped files. Once you download them, it'll look like that. When you double click on it, it'll try and unzip. It will ask you for a password. This password is in the email that TopMed sends you to say your results are ready. Okay, so you copy that password out, you paste it here, you say okay, and it'll unzip the file, and magically there'll be a chromosome 1 VCF for you. Okay. The other thing you need to do for tomorrow is you need to do your post-imputation QC. I will post the 2 steps for this. It requires you to install BCF tools. It's pretty easy to do. I will give you, I've given you instructions on how to do this as well. So it should be relatively easy. So the goal for tomorrow is to have your imputed VCF files that have been QC'd. Okay. So when I mean QC'd, I mean those 2 steps that I showed you about the allele frequency filter and the quality score filter. So you want to make sure we have allele frequency filtered, good quality imputation results to start with for tomorrow morning. Okay, so I will post all that information in the chat. We will stick around online until 3:30 to answer any questions. And then we will be back tomorrow at 9 o'clock. Please remember, we're not starting at 8, starting at 9 o'clock tomorrow morning. And we will then be looking at global and local ancestry inference. Okay. And thank you everyone for joining us for today. Good luck with the homework, and we will then see you tomorrow morning. Have a good afternoon and evening further. Bye-bye.